Everyone, and welcome to week 5 in environmental science. The first thing in our week 5 module is the content page. So this week we're going to be studying biodiversity and conservation. That's the content in chapter 8 in your textbook. So on the first tab we have some learning goals from chapter 8. There's some terms to know, and then some videos and worksheets to help you get to know the content. We will have a related discussion about wolf introduction and the Endangered Species Act. So this page has some information about the topic. Here's a peer-reviewed paper. Here's a video from the New York Times. Here's some Native American perspectives about wolf reintroduction. So get to know this topic and then make your initial post by Wednesday, and then make your follow-up posts by Saturday. This week's lab is a continuation of last week's lab. So last week you made a Burley's funnel and you harvested a leaf litter sample. That sample should have been drying, and hopefully by now you've collected some invertebrates in the alcohol that was underneath your funnel. This protocol is going to take you through trying to survey the invertebrates that are in the alcohol. It's going to require some supplies, maybe some tweezers, some kind of magnification so that you can see these bugs. Some of them are really, really small. Either a magnifying lens or a dissecting microscope. I have microscopes available for you to come and use in the lab. I'm also going to try to get Caitlin a dissecting microscope in the Academic Success Center on Thursday when she's there from 9 to 2. Hopefully you guys can stop by and take a look at your bugs then. An empty ice cube tray could be useful. Some little like white paper cups could work as well. These are just for separating out your bugs after you've identified them. This is how you're going to identify them. This is a web-based tool for identifying arthropods. So you're going to fill out this data table as you go through and see who's in your sample. The next thing you're going to do is do some analyses of your data set. You're going to calculate relative abundance for each of the types of invertebrates you found. And then you're going to calculate diversity, or DS, using this formula. This is a little daunting, but this example walks you through using that formula. So make sure you get this done by Saturday at the end of the day. I also want to point out that part of this laboratory assignment includes posting a copy of your data table in this leaf litter biodiversity discussion. So there's one analysis question that I'm asking you to answer that requires you to sort of look and see what everybody in the class found. So this is how you're going to do it. Everybody's going to post their data tables here so that you can see what others found. Let's go back to the modules view so we can get sort of an overview of what else is in the module. So we already looked at the content page, the group discussion about wolf reintroduction, the lab. This is where you'll post your biodiversity data table that you generate from the lab. Then, as usual, we have a practice quiz and a graded quiz based off of this chapter 8 content. And then the last part of this module is your plant population lab report. So a couple of weeks ago, you went out and you made a 10 meter by 10 meter area, and you sampled 10 random plots within that area. I gave you, this was the week 3 lab. I gave you guys some feedback on that. So make sure you take a look at that feedback before you write this lab report. So the lab report is worth 30 points, and there's a whole bunch of resources on this tab to help you write it. I'm using the same grading rubric, same guidelines. It should have the similar format to the last lab report you wrote with a title, an abstract, and then some kind of display of the data. I will say that I recommend that you put these data for the plant population, rather than graph them, I would put the metrics that you calculated for the population, like the number of individuals in the whole area, the density of individuals per meter squared, the variance, and that measure that you made of the distribution where you divided the variance by the mean. I would put all of those values in a table rather than try to make some kind of figure of the data set. So that's my recommendation for the data display portion of the lab report. So there's a whole bunch of information here to help you, and Caitlin and I are also available to help you. So please let us know if you have any questions about week 5 in environmental science.